Hello everyone. Welcome to this special webinar where we will be discussing projects related to infectious diseases research. In today's session, we'll talk about research projects developed for this program, Bioinformatics for Infectious Diseases. These include research proposals from the participants and updated resources on the following topics: origin and pathogenesis of the novel coronavirus, mutation frequency in the Ebola virus genomes collected during the 2014-2016 epidemic, linking drug-resistant variants in tuberculosis mycobacterium, enterovirus epidemic in children, and contamination of biologics production. This session is part of our Bioinformatics for Infectious Diseases online training program designed for biologists, clinicians, students interested in virology, microbiology, and immunology. For this program, we have collaborated with the Noguchi Memorial Research Institute for Medical Research at the University of Ghana. Today, we are joined by the research group at Noguchi, as well as the participants of the program. In the context of infectious diseases, we will also be discussing some of their projects and learn from their experience. Let us start by taking a look at the current progress of some of the participants who are here in the program, as indicated by the point system that we have for all of our OmicsLogic program. We can see how our participants are doing and completing this first phase of the program successfully. We started phase 1, which focused on the following topics: analysis methods and tools for genomic and transcriptomic data analysis of host pathogen interaction, methods to study relationships based on local and global multiple sequence alignment, phylogenetic analysis and evolutionary studies used for genome ancestry and population fitness, chemical and structural implications of sequence variation, mapped on PDB models, host pathogen interaction, and treatment response. Bioinformatics for Infectious Diseases is a 6-month online training program where we are learning about the use of bioinformatics and big data for infectious disease research. The program ends in the summer. After the first phase is over, we will go on to do an in-depth training in transcriptomics and genomic data. The next phase is going to focus on in-depth training in genomic data analysis. This will start on March 12th, 2021. The registration is now open and anyone with an interest can sign up. I am posting the link in the chat right now for the next phase of this training. So this program also comes with scholarship opportunities for students. The scholarships we offer are designed to help motivated, talented, and promising students benefit from special training and research opportunities. The criteria for approval include a reasonable need, indication of extra effort that the participant is willing to put into the program, and time commitment needed to go beyond the regular training. This will likely include keeping a regular record of topics covered in a detailed lab journal entry and participant in student-facing presentations about the learned topics. Now, let me introduce you to the program mentors and principal investigators. We have Dr. Harpreet Kaur, who is a certified OmicsLogic trainer and expert bioinformatician. She guides the hands-on sessions and assists participants with projects and analysis during the program. Her area of specialization is in genomics and machine learning. Dr. Mohit Mazumdar is the OmicsLogic project mentor and an expert in big data computational analysis. He has a PhD in computational biology and over 10 years of experience working with industry and academia. We also have Dr. Anita Gansha, who is a senior research fellow and lecturer at the Noguchi Memorial Institute in the University of Ghana. She is leading the projects as part of the Bioinformatics for Infectious Disease program. I will now like to invite Dr. Mohit Mazumdar to talk more about the upcoming program and his experience with research projects related to bioinformatics for infectious diseases. Over to you, Dr. Mazumdar.
Thank you, Bipsha, and hello, everyone. Let me share my screen real quick. Okay. So hello everyone. Yeah, so one of the major focus of this program, Bioinformatics for Infectious Disease Research and Training, is to support ongoing research projects that are spearheaded by Dr. Anita Gansha's group at Noguchi and her collaborators, having a long history of research in Ghana and other countries in sub-Saharan Africa to track, monitor, diagnose, and study the spread of drug resistance malaria, as well as identify novel approaches to for vaccine design, for as well as interactions between the malaria host, vectors, and pathogens to help conduct such research. So bioinformatics offers tools, data repositories, and analysis techniques that we are covering in this training program. So I've been working for the past several weeks collecting resources for malaria omics data. So today I wanted to just review the compilation of the data resources and the scientific literature associated with omics data for malaria research. So to compile this article, and you can see the link below here, and Beepsha, if you can share the link with everyone here as well. So to compile this article, we reviewed publicly available data sets generated by using next generation sequencing, and we looked into case studies that demonstrate the utility of sequencing techniques and how it correlates to the drug resistance in malaria by looking at multi-omics data, by looking at the entire system biology aspect, and then diving into the structural biology of the by looking at the protein and its mutations and the structural biology involved in it. So the objective of this review is to provide examples and starting point for students and researchers who are interested in bioinformatics for infectious diseases, and especially the genetic diversity diagnosis and for the treatment of malaria. So today I wanted all of us to take a look at some of the popular repositories and the resources listed in the post. So the first image that you can see here is European ENA, or the European Nucleotide Archive. So by searching the term Plasmodium, we kind of find out numerous results for omics data on omics data for this parasite. So this includes, like here you can see 170 assemblies and over 400,000 sequences and almost 34,000 experiments with 30,000 sequencing runs. So a reference of genomes, annotation of genes, examples of transcriptomic studies. So this is an invaluable resource for anyone interested in malarial research using the omics or big data. So second, another repository is PlasmoDB. It's very popular and is a biological database for genus Plasmodium. So the database is a member of the EU-PATH DB project, very, very popular project. So this data set and this database contains extensive genome, proteome, and metabolome information related to the malarial parasite. So another repository, this is here, the Malarial Host Pathogen Interaction Center. I mean, actually, it's not just a repository that they host, but this MAHPIC team, they strives to deliver unique large data sets, that holds potential for major breakthroughs that ultimately will advance our understanding for malaria and help diagnose, prevent, and treat the disease. So this initiative or innovation or innovative is a design of infectious disease studies, and they're using computational tools to integrate clinical, parasitological, functional genomics, proteomics, and lipidomics, and also metabolomics data. So all of these repositories and their links and even like the data sets are listed in that article that has been just shared. So then thinking about in the sense of transcriptomics analysis. So transcriptomics analysis of samples obtained in this human challenge studies can also help us deepen our understanding of the immune response with that symptom onset, allowing that characterization of innate immunity, and also that early gene signatures that almost always influences the disease outcome. So gene expression studies are actually helps and facilitates the identification of host factors that are contributing in the disease susceptibility. So one of the places, I mean, to look for data that can provide insights and answers related to gene expression of the gene, related to the expression of the genes that can characterize like this process of infection, is the NCBI Gene Expression Omnibus, GEO. So GEO is an international public repository that archives and is freely distributed. Microarray data is there. NGS data and other forms of high throughput functional genomics data are like regularly submitted by the research community. So these all information and much more is available there. You can look at. And then, as I was telling you, that next we looked into a case study. Uh, so this case study involving Plasmodium falciparum resistance to chloroquine. So as you, I think everyone knows that it's a former gold standard for anti-malarial drug, and now has a mutant form which is causing the drug resistance, basically due to the chloroquine resistance transporter gene, so PFCRT. So in this here on the left panel, you see that there is a bio project. So a bio project is a compilation which is available under NCBI. So here we found out about one data set that has been processed with this mutation that is uh, caused the caused the mutation at that position in amino acid. So it's leucine at 272 position, changing to phenylalanine, and the mutation was identified at cytosine at 814th position, changing to thiamine. So we traced that using the structure also, which is a cryo EM structure submitted in PDB, Protein Data Bank. But this data set we found out in NCBI, and they used, I think, blasticidin pressure, which resulted in this mutation at the coding region. So you can find out all these information in the blog post, and we are expecting that out of this resources, you'll be able to find some of the key questions that you have interest in. So one of the objective of this program, or this webinar, is to be able to help those who are starting in this journey. So we have prepared these articles and projects, and we are looking for research project ideas and proposals to leverage these resources and multi-omics data for noble insights into the spread of the disease, and as I told you, that prevention measures that can help address this pressing challenge across Africa. To talk about more, I mean, it makes more sense, and to talk about malarial research more, I would next like to invite Dr. Anita Gansha to talk about her research and the program so far. Thank you for this listening to me, and let me know if you have any questions related to the resources and the approaches and the multi-omics integration approach that we are taking, and then we are looking into the structural aspect. So I'll be happy to answer your questions. You can reach out to me at mohit@pine.bio. So I'll be also sharing that in the chat. And now I would like to hand it over to Anita to say a few words.
Thank you, Mohit. Let me share my slides. So, good afternoon to you all. Today, I'm not going to really talk about the studies I have conducted, but as we began this journey, I think a few weeks ago in January, we've been thinking about projects that we want to undertake to finish this first part of the training. And so at this point, some of the participants are ready to present studies that they are thinking about. This is just ideas that are still being crystallized. And so with time, you're going to develop it further. And to kickstart this, I have decided to look at investigating similarities and differences in host responses to SARS-CoV-2 and Plasmodium falciparum infection. And I'm going to do this with Dr. Bright Edu, and we are looking for others to collaborate, right? So a bit about malaria is that malaria, as Mohit has said, is a parasitic infection caused by parasites of the genus Plasmodium and transmitted by the Anopheles mosquitoes. The infection leads to acute and sometimes life-threatening diseases. And it is a major public health concern, although it hasn't got that much attention. In 2018, about 228 million cases of malaria were reported and 405,000 deaths. And most of these occurred in sub-Saharan Africa, with children under 5 forming the majority of both the morbidity and mortality. We all know the coronavirus disease, COVID-19, is also very infectious and a global pandemic, and everybody is interested in it. And it's caused by the severe acute respiratory syndrome, coronavirus 2, SARS-CoV-2. And when I checked last night, we had already had over 100 million cases and then 2.4 million plus deaths globally. Now, there are some similarities between malaria and COVID-19 symptoms, or the symptoms of the disease. Some of these include the fever, breathing difficulties, tiredness, and acute onset of headaches. And this may lead to misdiagnosis of COVID-19 for malaria, especially in the malaria endemic regions, and vice versa. This particularly is seen where a lot of the malaria diagnosis relies on just the symptoms. I can speak for Ghana and a lot of African countries. We self-diagnose and go to pharmacies and get our treatment based on these symptoms. So it's very likely that we have missed a lot of the COVID-19. Now, as a result, and many, many more reasons, we are sure, there is a low prevalence of COVID-19 in malaria endemic countries. And this is what was not originally expected, considering the fact that these two infections may have similar symptoms, and you can imagine the co-infections with COVID and malaria may be complex. So some of the reasons that have been assigned to the low prevalence of COVID-19 in malaria endemic regions are the age structure, there's a lot about the fact that COVID-19 is most prevalent in elderly people, and the age structure in Africa is more youthful than in the Western world. So people over 80 or above 65 who seem to have other underlying conditions are most affected by COVID-19. But we've seen that the age bracket for COVID-19 is lowering with this second wave of the infection. But there's a whole area that is still being unraveled. Now, as I've said, there are similarities between the COVID-19 symptoms and that of malaria. So you can imagine that the immune responses that people who have been repeatedly infected with malaria may be playing a role in preventing very symptomatic or severe COVID infections in the malaria endemic regions. And some of these roles have been assigned to the ACE2 in malaria and COVID, basically because the angiotensin 2 that is being catalyzed by the ACE is found both in COVID-19 and also protecting malaria infections. And then the role of interferon and the neutralizing antibodies in malaria and COVID-19 has also been postulated to contribute to the low prevalence. And the use of hydroxychloroquine and chloroquine in COVID-19, there's a lot of controversy around that, but this may also contribute to the low prevalence as we keep treating people with these drugs in sub-Saharan Africa or malaria endemic regions. But we also know that the co-infections with malaria and COVID-19 the interactions can or would expect it to rather worsen the disease phenotype. So the problem that I think we have to unravel is the fact that even though we have significantly lower COVID-19 in malaria endemic regions, we may be expecting, or we're expecting co-infections to be more harmful. And although the role of the ACE2 receptor, interferons, and neutralizing antibodies have been postulated to be involved in a low incidence of COVID-19 in Africa, the full spectrum of the host similarities and differences in response to both infections has yet to be unraveled. So we are proposing to conduct a comparative differential gene expression analysis on available RNA-seq data on the host cells infected with Plasmodium falciparum and SARS-CoV-2. The overall objective is to elucidate the significant similarities and differences in gene expression profiles and the underlying biological and molecular mechanisms. Specifically, we want to conduct a differential gene expression analysis on SARS-CoV-2 and malaria-infected human cells, red blood cells, and there are other cell lines too. And then we're going to control for transcriptional features shared by SARS-CoV-2 and other viral infections such as SARS, MERS, and H1N1, just to make sure that we are dealing with COVID-19 in our analysis that will determine the similarity between the transcriptional features of SARS-CoV-2 and malaria-infected cells. And when we have found these differentially expressed genes between these two diseases, we will conduct gene ontology analysis to look for the significant molecular and biological functions involved in the host responses to SARS-CoV-2 or COVID-19 and malaria. We are expecting to obtain the full spectrum of genes and pathways involved in both COVID-19 and malaria, and then to describe the molecular and biological functions, underlining the similarities and differences in the host responses to COVID-19 and malaria. And then we look at the potential synergy between these host responses to COVID-19 and malaria. So we have data sets from 3 studies. One is looking at repeated infections in children, the transcription or transcriptional profile of children who have been repeatedly infected with malaria. And then the other one, study 2, has got the transcriptomes of individuals and cell lines infected with SARS-CoV-2, MERS, H1N1. And then the third study has got red blood cells infected with malaria Plasmodium falciparum parasite and the transcriptome analysis of that. And these are the references. Thank you very much.
Thank you, Anita. I also have a question. Is anyone else from your group ready to present? Because I think I need to make them co-host to do that.
Yes, I think that Courage and Nigel will also present their work. And Ellie.
Okay, so I've made,
okay,
I've made Courage the co-host next. So Courage, if you would like to share. And about this, thank you for the presentation, Anita. It was really interesting. I wanted to ask, there seems to be a clinical trial that is just getting started. Are there cases that you know of that have both malaria and COVID?
You mean places? I mean, you would expect to see that in sub-Saharan Africa. Because,
are you aware of cases where people actually got both diseases at the same time?
Well, I haven't really looked at that, but I'm sure Bright, who has all the data, can tell us that unless they haven't tested, but the samples are there and they can be quickly screened for malaria. Yeah.
I see. Okay.
But it must be very common. Yeah.
You think it is common?
Yeah, because about 20-25% of people in at least Ghana are asymptomatically infected with malaria. So it's very likely.
Okay, great. Thank you very much.
Hello, everyone. I hope you're all doing fine. Can everyone see my screen?
Yes, we can see it.
Okay. So my name is Courage Adobo, and I'm proposing a project for these courses we are taking. Genetic variation studies of sexual stage-specific antigen PFS 230 and PFS 48 and 45 of Plasmodium falciparum population in Ghana. Yeah. So we know malaria is caused by malaria caused by Plasmodium falciparum is one of the most deadly form of malaria, especially in sub-Saharan Africa, affecting millions of people and especially children under the age of 5. So control interventions against malaria has seen the use of anti-malaria drugs like artemisinin and previously chloroquine and stuff. And then there are also other vector control measures. But what is lacking among these tools is a broadly effective vaccine. The currently most advanced malaria vaccine, RTSS, has shown low efficacy in clinical malaria. This low efficacy studies have shown that is largely due to the genetic diversity of the targeted antigen, which is the circumsporozoid protein. Yeah. Now, transmission blocking vaccines will induce responses against the malaria parasites, gametocytes, sexual stages, thereby blocking fertilization and halting reproduction. Two antigens, PFS 230 and PFS 48 and 45, which are expressed on the surface of gametocytes, have been shown in research papers that they elicit strong immune responses. However, genetic diversity of these antigens have not been fully explored in the Ghanaian parasite population. Okay. So the problem is the efficacy of current malaria vaccines are hindered due to the diverse nature of the targeted antigens. To have a successful transmission blocking vaccine, it will have to induce the immune responses against the malaria parasites at the gametocyte stage. However, the genetic diversity of transmission blocking vaccine antigens have not been fully explored in the Ghanaian population. So there is the need to therefore explore the genetic diversity of this transmission blocking antigen to further estimate how effective vaccines will be if they target those antigens. So the objective of the project is to scan for genomic regions inside these antigens and then find variations in them in the population, and then identify the types of mutations that these variations cause, and further see whether these mutations, the functional effect they have on the translated proteins. So I'll take my data set from malaria gene, the public, the community projects, and this is the URL to the various data sets. References. Thank you.
Courage, I just had a question. Were you going to work on this on your own, or is there anyone else that you're planning to work together with?
So I've not gotten any partner yet. So for now, I'm working on it alone, but I'm looking for partners.
Okay, so you're open for others to join you if they're interested,
Hello.
right?
Yes.
Okay, and I guess at the end of this session, we can talk more about what it would be an effective way to partner on these different project proposals. Thank you. If anyone has any questions or comments on this, please feel free to add those. And if for anyone else that's going to be presenting, if you could just start off with just a few words about yourself, your background, kind of, how you ended up getting interested in this project, I think that would also help others consider how they may contribute. So, if your background is in bioinformatics or your background is in something else, I think it would be more easy for people to see what they can contribute. Thank you, Courage. So I assume the next person would be Benedicta.
Can someone else present? I'll present, please. I'm not ready.
Okay, who else is ready to present?
Nigel, I think.
Yeah, Doc, I'm ready to present. So let me share my screen.
Okay, Nigel, hold on. I think I need to make you a co-host. 1 second. Okay, Nigel, I made you a co-host.
Right. Thank you very much. So let me share my screen. Hello, everyone, and my name is Nigel Dolan. I'm a research assistant at the Noguchi Memorial Institute for Medical Research. And my background is in bioinformatics. I'm working under Dr. Anita Ganta, who is my supervisor or my PI. And my background in bioinformatics entails computational drug discovery into neglected tropical diseases like Rowley ulcer, tuberculosis, leprosy, and then also malaria. And so this project that I'm thinking about is regards to malaria, and it's a computational guided approach towards the discovery of natural product compounds resistant to PFMRD1 and PFCRT. These are two gene mutations which have been known to confer artemisinin resistance in Plasmodium falciparum. So the background. Malaria, which is a protozoan infection, is caused by Plasmodium parasites, and this remains a major cause of morbidity and mortality worldwide, primarily among children less than 5 years old. And yes, in 2018, it was estimated to have caused around 228 million cases globally, of which 93%, which constitutes 113 million, occurred in Africa, according to WHO. Now, the map you can see shows P. falciparum malaria parasite prevalence in Africa and the percentage of children infected with P. falciparum. So in blue, blue shows the low areas where we might see 1 or 2 infected children out of 100. While in high areas, which are indicated by red, it might be more than 50. Now, this alone demonstrates how entrenched the disease remains throughout much of West and Central Africa and parts of Southeast Asia. However, this is not all gloom, as there have been therapeutic gains in trying to curb this menace. The recent and current frontline therapy is the artemisinin-based combination therapies, or the ACTs, and these have been invaluable in the substantial decline of the burden of malaria. The challenge. Yet again, however, the two main pillars for malaria control and beyond remain targeting the Anopheline mosquito vector and effective case management, which is crucially dependent on the efficacy of the deployed anti-malaria drugs, herein ACTs. Yet there is a growing concern that serves to question the efficacy of this therapy. There have been reports of treatment failure of ACTs in Southeast Asia, and this parasite is just relentless, I must say. Antimalarial drug resistance in Plasmodium falciparum tends to emerge in low transmission settings, in particular in Southeast Asia or South America, before expanding to high transmission settings in Sub-Saharan Africa. Arrival in Sub-Saharan Africa of these very difficult to treat parasite strains can have a huge impact on malaria morbidity and mortality. And yes, they have arrived. The cause, mutations. In 2014, reports established that resistance to artemisinin is due to the Kelch propeller domain of the parasites. However, of importance to this study are the PFCRT and then PFMDR1. So alleles of Plasmodium falciparum chlorine resistance transporter gene, which is the PFCRT, have been associated with chlorine resistance in Plasmodium falciparum. And then the Plasmodium falciparum multidrug resistance, that's the PFMDR1 gene, is also associated with both chlorine and amodiaquine resistance. So you will see in this image here that the parasite, which is shown in gray, a gray oval, is shown within the red blood cell, which is in red. The DV, which is the digestive vacuole of the parasite, is a compartment within a parasite where the catabolism of hemoglobin from the host red blood cell occurs. The breakdown of hemoglobin results in reactive heme, which is toxic to the parasite, undergoes detoxification to become hemozoin. Now, medications from chloroquine class bind heme and interfere with detoxification. Yeah. But PFCRT and PFMDR1, which are DV or digestive vacuole membranes proteins, it's thought that the PFCRT transports drug out of the DV, while the PFMDR1 transports drugs into the DV. Now, the mutation in the PFCRT is essential to chlorine resistance, while that in the PFMDR1 is essential to augment also chlorine resistance. A mutation in these transporters have been found to mediate a resistance of artemisinin, and thus the goal. This project therefore seeks to virtually screen multiple natural compound libraries from different databases. First of all, to identify compounds which could potentially have binding affinities against both the wild and mutant PFMDR1 and PFCRT. Now, to what end is this? The end of which is to identify compounds which could potentially circumvent the resistance of mutant PFMDR1 and PFCRT, so that at the end of the day, we could have new compounds that could potentially be moved to become drugs that could address the issue of resistance in regards to this parasite and the menace which produce malaria. And so these are my data sets and my references. Thank you very much.
Okay, great. Thank you, everyone. Sorry, thank you, Nigel. Would you be also open to others contributing to your project?
Yes, please, I'll be open to others contributing. Yeah.
All right, excellent. Thank you very much. Again, the process for anyone to contribute to these projects, either from the participants of the current program, you know everyone involved. For those who are not a part of the program, we're going to discuss the upcoming program on genomic data analysis, as well as the research fellowship. If you're a part of the research fellowship, we can also probably match you with somebody who you're interested to contribute to, and it will be up to them to decide if they're open to that contribution. Who would be, are there any questions, sorry, for Nigel? Okay, if there's not, who would be the next person presenting?
Hello, I have a question.
Yes, go ahead.
Yes, my name is Ayo Fayel. So my question, thank you, Nigel, for the wonderful presentation. So I was just thinking, your project is trying to look for compounds that will inhibit those two genes, both the wild and the mutant genes. Now, how are you going to get data that will show, that will have an array of compounds to test? Since you are looking for compounds, or is that are there data that has all of the possible compounds which you can actually test for? Thank you.
Thank you very much, Ayurinde. Something else I forgot to add to it. So this study is also comparative, because at the end of the day, I would be looking at the binding affinity of those compounds with regards to artemisinin. And so how well they do determines whether or not we are going forward with those particular compounds. And then so Zinc database, I forgot to mention, part of the data set, Zinc database has a catalog of compounds, which, and the subset of which are, some of them are from Africa. So you'd have AfroDB, which has a catalog of a number of compounds. And so my research is targeted towards compounds that are from Africa. that you are going to screen against these genes, then see the ones that perform better than artemisinin and go forward with it. Yeah, so this is just a clearing study. Yeah, thank you.
All right, great. Thank you. Benedicta, would you like to go next?
Yes, please.
Hello, Ilya. I also have a question for Nigel.
Okay. Just for the sake of time, if we could move on to Benedicta, but you can post your questions in the chat, and
Okay, no
we will
problem.
make sure to share the contact details so that for more questions after that. Okay.
All right. Thanks. Thanks a lot.
Okay. Thank you at that time. Go ahead, Benedicta. Okay. Benedicta, are you able? We cannot hear you or see your screen. Okay. Benedicta, can you still hear us? Okay, maybe it seems like
He lost the connection, I feel.
yeah, lost the connection. Okay, so maybe we can continue and she will join back. We also wanted to explain a little bit more about how these projects are formed and how anyone can get started with a project even without having a lot of information on bioinformatics and other things. I see Benedicta joined back. Benedicta, can you hear us? Okay, so I don't know if
Ilya, I can take it from here and introduce everyone to the research fellowship.
Okay, great. Thank you, Bipsa.
Thank you. So as many of you are interested in collaboration or looking for opportunities of how to work on projects like this, we have this research fellowship program. So I want to thank Dr. Gansa, Courage, and Nigel for presenting. We are looking forward to seeing their projects fully fleshed out. For the remainder of the Bioinformatics for Infectious Disease program, participants of the training and our other research fellows will be collaborating together with our mentors and experts from the Tauber Bioinformatics Research Center on completing the analysis, and to give everybody a sense of how the research component of the program works, I'm going to be sharing a few more slides about the research fellowship program that we have. So one of the main challenges that we have seen for many researchers, students, as well as research fellows who are starting their bioinformatics work is to prepare a research plan that fits with the type of data and leverages big data analysis tools effectively. So we have a step-by-step process which guides our research fellows and helps them understand the logic first and then apply it to their own research question. So as a result, anyone starting on this journey in bioinformatics can start developing a project of their own. In fact, we have already worked with many students who have completed the training and developed interesting research projects which have won awards and even got published in peer-reviewed journals. So here are some of those projects by our graduated research fellows who have worked on these different areas of bioinformatics research. Yes, and for all of you who are showing your interest in the chat box, please put your email ID down, or please email us at marketing@pine.bio or africa@pine.bio. So we also have a team in Africa who support us, who support the Pine Biotech team. So you could also reach out to them. And thank you, Fiza, for sharing. So as I mentioned, many of the past participants had the chance to present their analysis in front of their classmates or at a conference, in poster sessions or workshops. So here is an example from one of such conference that we had in Baton Rouge in Louisiana. So where we presented on different topics, and here was a presentation by our team. So to help participants reach this stage of a complete project that they can then present to a larger audience, we work with them through the steps of the project design. So we have these different steps that we have in the project design process that we work with each one of the participants. So each research fellows goes through this process, starting with learning about the different multi-omics topics, then completing certain tasks related to them, then starting their literature review on an area that they are interested in researching on, working to find data sets which would be apt for the kind of problem that they are trying to solve, and finally going forward with their analysis, their presentation, presenting to an audience like we have today, with a wider audience of people, getting it reviewed by our experts at the Tauber Bioinformatics Research Center, and then finally taking it to a stage where it can become a publication like you see here. So this is a very step-by-step process, and the research fellowship is offered for a period of either 3 months minimum or a period of 6 months. It can go in longer. But what we aim to do is that we start this process by training each research fellow, making them familiar with the topic, making them familiar with the research that is going on in the different domains. So it can be anything. It's not just infectious diseases. You can be interested in oncology. You can be interested in precision medicine, like you see this project here. So there are a lot of opportunities for everybody interested in different topics. We also have a working group right now who are working on Alzheimer's. So there are these different opportunities available for everyone that you can explore as part of the research fellowship process. And for example, this is a project that was developed by Dr. Gansha in collaboration with Dr. Emeka Patrick Chukwuka in part of the research fellowship program, where they were looking at the analysis of transcriptome profile of SARS-CoV-2 infected cell lines to understand the mechanism of action of repurposed drugs for COVID-19 treatment. So as you see, Dr. Gansha has already spoken about how we are looking at the different prevalence of COVID-19 and malaria across regions in Africa. So this was one of the projects she worked on related to COVID-19. So now, to help those who are starting in this journey, our team has prepared several advanced research projects that anyone can take to learn more about genomic and transcriptomic data in application to various pathogens. I would like to invite Dr. Kaur to present some of the new projects that are available right now on our platform. Dr. Kaur, over to you.
Thank you, Bipsa. So, hello everyone. My name is Harpreet Kaur and I have a PhD in bioinformatics. In my current role, I'm OmicsLogic bioinformatics trainer with the Pine Biotech. So here I'm assisting participants to learn about data analysis, machine learning and visualization in the application to the large data sets used in the biomedical research. So besides, I'm involved in designing different research projects using publicly available data with our team members at Pine Biotech. Here we are continuously designing different research projects using publicly available data based on the studies published in high impact journals. Thus, we have collection of projects where you can learn how bioinformatics can be applied in the field of infectious disease using publicly available NGS data. So the first project here is on Ebola. So as we know, Ebola virus causes a deadly Ebola virus disease in the human, which is also sometimes referred as Ebola hemorrhagic fever. So 4 out of the 6 known Ebola virus cause life-threatening viral infection in the humans, which is also highly contagious. So the recent epidemic outbreak in West Africa has raised concern about the implication of this virus on a human population. So here we have designed a dedicated project on Ebola based on the original publication data to understand its origin and to find out what are important mutation or the variants that are for this particular malignancy. For this particular outbreak. So during the 2014 to 2016 outbreak of the Ebola in the West Africa happened across different regions. The next generation sequencing can be used on the large scale to track the development of this epidemic. So here we used genome sequence data from the Ebola virus strain to perform MSC and the phylogenetic analysis. So we used multiple sequence alignment and phylogenetic analysis to determine the evolutionary relationship between these genomes. So we have identified a group of samples and how they are related to its phenotype. So the phenotype that we choose here in this case is its geological location, that is the country from where it is originated, like Gabon, Liberia, or UK. Next, we also see how we can analyze multiple sequence alignment and nucleotide label and amino acid label to understand the differences and the mutation among different samples. So we will learn what are the key mutation in a specific region that result alteration in the pathogenicity. So we also analyze mutation between groups of the sample using comparative analysis to study mutability. So this method helped us to identify the origin of polymorphism, which is believed to a cause of an increase in the ability of virus to enter the host cell. And the next project is on COVID-19. So when this COVID-19 pandemic outbreak happened, so there are the several questions arise that needs to be addressed. So these questions can be like from where it is originated or from which animal or from which location it is originated. So how it start infecting humans. So for this purpose, to understand this, we have designed a project on COVID-19, that is origin and the pathogenesis of novel coronavirus. So here first, based on the multiple sequence alignment and the phylogenetic analysis, we will try to understand its genetic origin, that is from which animal it has been originated. So in this, you will learn to perform phylogenetic analysis to understand the origin, whether how it is associated with the other strains of COVID-19 as well. So then multiple sequence alignment will help us to understand what are the major differences and the variations taking place between the different strains of the COVID-19. So in the next part, we will also learn how and we can obtain what are the different key components or the protein components in the virus and the host that are responsible for its pathogenicity. For instance, ACE2 receptor binds to spike protein. So how you can visualize the structure of this protein and its complex with the ACE2 spike ACE2. So how you can understand the differences between the different regions in the structure. So we have incorporated this part as well. In the next part, we will also have tried to incorporate transcriptomics data analysis to identify a signature that can discriminate the sample that are ruxolitinib treated and untreated samples. So here we have employed different data exploratory analysis approaches like differential gene expression analysis, principal component analysis, and hierarchical clustering to understand what are important signatures that can distinguish these classes of the samples. Then the third project is about tuberculosis. So here we have designed this project to understand the drug-resistant variant in the mycobacterium. As we know, 1/3 of the world population has been infected with tuberculosis. So this tuberculosis is caused by mycobacterium tuberculosis. So this disease has severe symptoms including chronic cough, blood present in the sputum, then the weight loss, etc. But the major concern with the tuberculosis is the drug resistance in the mycobacterium. So there was a research work was published in one of the top journal, that is in Lancet. So where authors have performed whole genome sequencing for the prediction of the mycobacterium tuberculosis drug susceptibility and resistance. In their study, authors sequenced a training set of nearly 2100 mycobacterium tuberculosis genome. So we have taken this data. So in this project, we have employed various GWAS approach to identify those genes that are associated with the drug resistance. Here mainly we used gamma, Plink, and the slope methods. Then the next project is about enterovirus D68 outbreak. So one of the diseases that is acute flaccid myelitis, which is a severe neurogenic disease in the children caused by this EVD68 virus. So this AFM disease is like a polio-like, defined by the acute onset of flaccid paralysis in the setting of spinal MRI demonstrating a longitudinal lesion in the gray matter of the coat. So in this project, we have tried to understand the evolutionary relationship between the strains from various outbreaks of EVD68, which are occurring at different time points based on the NGS data. So it is first emerged around 1968, but in the last decade, this outbreak of EVD68 has been reported across the different parts of the globe, particularly in the US and the Europe, like after every couple of years, like 20 recently it was happened like 2012, then 2014, 2016, and so on. So they have also observed the pathogenicity of this particular virus is actually varying among different outbreaks. So here we have used multiple sequence alignment as well as various phylogenetic analysis using different phylogenetic tree reconstruction algorithms to find out whether EBD strains from the different outbreaks at different points are the same or the different based on the phylogenetic and multiple sequence analysis. And further, we have identified how they are different based on the phylogenetic analysis. Then the multiple sequence alignments indicate there are number of the variations occurring in these strains, but not all the mutation transferred at the amino acid level. Still, there are like 2 to 3% of the variations they taking place. So participants can explore all these projects and learn about how you can employ various bioinformatics approaches to analyze NGS data from various infectious pathogens. So that's all. So now I would like pass this session back to the Beepsa. So over to you, Beepsa.
Thank you so much, Dr. Harpreet. So I want to take this opportunity to invite you all to see where the projects that Dr. Harpreet just introduced is on the platform. So if you are again joining us for the first time, please come with me to edu-t-bioinfo. I will be posting this in the chat box right now. So once you are in this platform, you will notice, so I'm already logged in. So you'll find, if you're not logged in, either a register or a login button. And you can register with either our email ID, or there are also registration via any of your social media IDs. So please go ahead and register. And if you're already on the platform, please put down number 1 in the chat box so that I know that at least some of you are there in the platform and you can explore the resource that I'm showing along with me. So please put number 1 in the chat box. Thank you, Emmanuel. Anybody else who is on the Edu platform? Thank you, Martha and Courage and Elizabeth. Anybody else? Thank you, Nigel. So we already have a lot of you who are in the platform and also part of this training program. Okay, thank you, Saida. So let us explore the first free resource. Now, I know that many of you who are present in today's meeting have already taken this course. But for anybody who is joining us for the first time, we recommend you to take the Introduction to Bioinformatics. And the reason is this course introduces you to the different opportunities available. And we encourage everyone to take the course so that you can go ahead and take the intro survey, which gives us an idea about who you are and what your interest is. So this helps us to collaborate with you on your area of interest and provide you those opportunities which are available in the area you're interested in. So the next step is to find the project. So if you note in the projects, if you click on this column, it will open up to this tab. So these are the projects that Dr. Harpreet has been showing you. So let us see the projects which are there. So we discussed tuberculosis, COVID-19, the Ebola project, and here are the direct links. So some of these projects, like the COVID-19 project, is free and available for everyone to start taking the project. So I'm posting the direct links, but you can again go from the project section right here and you can see more, or you can just click here. It will lead you to this project page. And as I mentioned that these projects are not just on infectious diseases, there are more projects on different areas. You have projects also on cancer and oncology, which are here. So please feel free to explore these projects. And if you want to study what other participants have done in the previous program, I'm going to be sharing with you a link to the project examples. So here is the link to project examples from our previous participants who have taken part in such training programs and have successfully done a project. So most of these participants who have been featured in this project example page are our research fellows. So they have done some of their work and it is featured here. Many of them are going towards working on a publication or working on writing a paper. So these are the different projects that you can see, and they cover different areas and applications of bioinformatics to different kinds of data sets. So please feel free to explore this page as well. And as for the next steps, like what are the different options that you have to join a program like this? So as I mentioned earlier, so you have already the upcoming genomics program as part of the next step or the phase 2 of the bioinformatics for infectious disease program. And this is open for registration if you are interested. We have already also mentioned the research fellowship. The next program which is available is the OmicsLogic transcriptomics program. So this is a program completely on RNA-seq data analysis. So if you are interested in learning more about NGS data, then this is the program for you. The other program, which is ongoing. So this program has already started on February 16th, but you can still enroll for the program if metagenomics is an area that you're interested in learning. And this program is also a collaborative program with an institute in Nigeria called the Helix Biogen Institute. So this program had only 2 sessions so far. It's open for registration. So you can also join this program. So we are covering different omics topics as part of these programs. And another program which is starting very soon from March 15th is a program on precision oncology. So we are going to be studying example data sets and talk a lot about cancer and the role of precision medicine in understanding disease like cancer. And here are the different links to enroll in these different programs. So let me quickly share with you the links. So I believe that there is a lot of links. If you are interested in something particular, please let us know. I'm going to be sharing all the pages where you can actually enroll for each of these programs. So please feel free to log into these pages and enroll for the program. So I think
Thank you.
this is...
Thank you, Beeps. I just wanted to mention to all of those that are already participating in the infectious disease program, you have until the end of today to finalize your submissions of your project proposals, because we will need at least 3 days to review all of the project proposals with our collaborators at the Tabor Bioinformatics Research Center and with Dr. Gansa, so that we can finalize it by our next session on Friday. And if you have any questions about your project proposal, if you are finding it difficult to find a data set, or you don't really know if there is something that you would like to focus on, consider joining one of the proposed projects that we saw today from Nigel, from Dr. Gansa, from Courage. I don't know if Benedicta is still wanting to present her project, but those would be projects to consider to contribute to. And then also one of these projects described by Dr. Kaur, who showed these are projects that you can start with, but then you can expand on those by adding additional data sets or trying some of the other example methods that you've learned about in the program that we are doing right now. So Benedicta, would you still like to share?
Yes, if you can hear me now.
Yeah, we can hear you. Great.
Okay. Can you see my screen?
Yes, we can. But you probably should change the screen because we see your presenter view.
Okay.
You can change it right above where it says display settings. You can just do. Yeah, you see it says swap. Yeah.
Okay.
Now we see it.
So the title of my project is looking at the structural patterns of diversity and selection of Plasmodium falciparum apical membrane protein 1 gene in Ghana and also across multiple populations in the world. So my name is Benedicta Mensah. So I have a background in biochemistry, epidemiology, and biostats, and then somewhat knowledge in biostatistics, in bioinformatics. Sorry, I still have biostatistics here. And my research interest is in molecular and genetic epidemiology, more specifically looking into infectious diseases and non-communicable diseases such as cancer. So this project is an extension of my PhD work, where we did sequence the apical membrane protein. And a little background into that is the apical membrane protein is a transmembrane protein that is released into the surface of the merozoite protein during invasion of merozoites into erythrocytes, that's the RBCs. And some studies have shown it's been under selection by the immune system, and is one of the primary targets for malaria vaccine and development. And to effectively prevent malaria and subsequently eradicate, there's a need for us to develop an effective malaria vaccine. So the aim of this project is to look at the genetic diversity of the AMA1 gene and whether this is under balancing selection or immune selection, and also to look at the structure of the PFAMA1 gene and to see whether the variations that we see in Ghana and also variations that we see in other parts of the world like Southeast Asia and Eastern Africa, to see whether this affects the protein structure and also functions such as immune selection. So the project summary involves looking at diversity indices such as nucleotide sequence diversity, haplotype diversity, the amino acid sequence diversity, and also look at the protein structure and its folding, look at FSD and genetic structure, for instance, looking at PCAs to see whether these genotypes cluster differently for the different populations such as West Africa, and then also some other parts like Southeast Asia, and also to determine the evidence of selection using Tajima D, and also from this course I've learned how to do the 3D profiling of protein structure. So that's what this main project I would be focusing on, to see whether differences in these nucleotides affects the protein structure, and whether this also affects how these different genotypes would functionally select by the immune system. So I will use data that we have generated here at Noguchi, NGS data, and also data from a project that's the malaria gene, where they sequence the whole falciparum genome for multiple countries. So I would extract data I will extract the AMA1 gene data from this project and then compare to see the indices that I have indicated, to see how they differ between the Ghanaian populations and then also other populations across the world. Thank you.
Thank you. And so can you just describe a little bit better specifically what type of data? You mentioned you have your own data, but there's also this publicly available data.
So what exactly what type of data is this? So it's a genomic data of Plasmodium falciparum gene.
Just that one gene, so it's amplified for that gene?
No, it's the whole genome of falciparum. So I'm gonna extract the AMA1 gene and then compare.
And what kind of metadata is available for that data? Is it just resistant, non-resistant, or is it also resistant to different types of drugs or anything else like this?
So as resistance to, this is a malaria project. So it sequences the whole genome and then you can decide. So I have the link to the project, but I am interested in looking at the AMA1 gene. So I'm not sure what kind of metadata they have there. But I'm sure there's a sequences of the whole genome. So.
Okay, and are you interested in others potentially contributing to your project?
Yes, please, if anyone is interested.
All right, great. Thank you very much. Anita, did you want to add anything to that?
Yeah, I was going to say that the Malaria Gen project does not really have available metadata, just the location of the samples and sometimes the level of parastemia. But if one is interested, then you have to apply, and then you know the resistance status or information about the clinical study that went with the data.
What are you people doing? Official.
Thank you.
मैं तो एक वेबिनार जो अटेंड करता था.
Sorry, not sure if somebody is speaking. Okay, thank you for that clarification, Anita. So if anyone has any questions about this project or how to join, I hope that we were able to answer all of those questions. And again, just to kind of summarize this webinar is a part of our ongoing program. This program is designed for research, kind of transitioning into research by those who have received the training that we have. And we also have a number of other programs and resources that Beepsa mentioned. So Beepsa, I'll pass it on back to you to wrap it up.
Thank you so much, Dr. Mensah, and thank you, Elia, and also Dr. Gansa. So as we are ending today's session, I want to ask if you have any questions. And as you see on my screen right now, I'm sharing our LinkedIn page. So this is also a group where we collaborate with a lot of participants from all over the world. So please feel free to go ahead and join the group. I've posted the link in the chat box, and we also have a similar Facebook group here as well. So any of these social media channels that you feel comfortable in, please go ahead and follow us. And you will also find snippets of what we do shared in our Instagram page at Bioinformatics Pine. So you're able to find us on all of these social media channels. So please continue to follow us in any of these places. And we hope that you not only follow us, but also gain something out of this. We share interesting research articles from time to time. We talk a lot about our programs. We talk a lot about the job opportunities which are there, especially on our LinkedIn and our Facebook groups. So please feel free to take advantage of this opportunity and see if you are also able to share with our other members something that you feel interested in, and you can have a conversation. So what we intend with our community here at Pine Biotech is that our bioinformatics community can not only learn from a program like this or collaborate on projects, but they continue to learn from each other and collaborate with everybody who is present in that group. So Fiza has a question. Are all the projects discussed here a part of the bioinformatics research program? I mean, do I need to register for every course individually, or overall registration is required? So all the projects that are mentioned here would be available as part of the research fellowship program. So what you need to do is that when we start the research fellowship program with you, you would need to specify a particular area. Now, there are a lot of projects that I have shown you. Say, for example, there was this Ebola project. So if you're particularly interested in one project, we would be working to make sure that you are learning all about Ebola. Now, you can definitely do other projects, but what we try to do as part of the research fellowship or any of our training programs is to make sure that you're focused on one particular direction. Because firstly, as I mentioned while I was presenting, that it's not easy always to come up with a particular form of analysis, find the right data set. So those are some of the struggles that we see every research fellow, notwithstanding what their educational qualification is or how much they know, they struggle with, because these are not easy to come up. So you are going to spend a lot of time on one particular topic. So yes, you can choose any of these research projects. In fact, the projects that we mentioned as part of the examples, all of those projects are novel research projects, which means that we have not proposed those topics. So the projects that you see here have been proposed by these students or these participants who have taken part in the research fellowship program. So they propose the projects, and they can only propose the projects once they go through this entire process of training, learning, going through the different example projects, and then understanding that, okay, so once I've learned from here, what are the different opportunities to explore this in a different research question. So that is exactly what the research fellowship is about. So you can choose any of these topics. Now coming to Ayorinde's question, can one still join this program despite having started since January and would end in a few days? So Ayorinde, as we mentioned, that this is a 6-month long program, which we are doing with Noguchi. Now the first part of the program, Bioinformatics for Infectious Diseases, is going to be over. You cannot join that program. Now, the next program that we have and you can join is the OmicsLogic Genomics program. So this is phase 2 of the program, where we are going to be talking about genomic data analysis in respect to bioinformatics for infectious diseases. So it's a continuation of the training program. And the next, again, the training that we are going to be talking about is transcriptomics. So you can still join this program, which is going to be starting on March 12th. It is completely open for registration. So you have not been a part of the first part of the training, but as if you see this program and its syllabus, it starts with a very introductory session. So we have broken down this entire 6-month program into different parts, and you can still take part in this program, as we are going to be starting right from, like, you can say stage 1 and building up your understanding of genomic data and how to work with that kind of data. So here you can find the different syllabus for the program. So you can see the different dates which are there, the different topics that will be covered on each of these dates. So you can definitely join this program again. So do we have any other questions that you want us to address right now? Because we are almost close to ending today's session. And again, if you still have some questions, and if you do not have them right now, please feel free to reach out to us at marketing@pine.bio. So, okay, so for Emmanuel's question, which is about metagenomics. So metagenomics started, I mentioned, on February 16th. So we have had only very 2 introductory sessions, and our next session is not until tomorrow. So tomorrow is going to be like the 3rd session. So we have just started. So if anyone is interested, we welcome you to go ahead and join the metagenomics program as well. So this is like, I feel it's a wrong link. So if you have put in your email address, I think Emmanuel, you have already shared with us the email address. We will be sharing with you the details. And as I mentioned, we also have a team in Africa. So let me share with you the correct address. I think Adi Tayo has also shared it in the chat box. So you can also reach out to the Africa team at africa@pine.bio. So if you go to the, okay, let me share the email ID again in the chat box. Right. So what I wanted to show you is that we have had only this introductory session and this session on microbiome as a functional bacterial community. And the next session is going to be tomorrow. So if you're interested, please go ahead and join. And if you need more information, you can reach out to either of these email IDs. So you can reach out either to me or to Aditayo at africa@pine.bio. And thank you, Aditayo, for sharing this. So I hope that this is all clear to you. We are having several different programs that suit different research interests. So you can reach out to us. And I hope that with all of the different programs available, you are not overwhelmed, because if you are, You are, again, can ask us questions, and I think we will be able to help you address some of those questions. So we had a question regarding like data science program. So we had just concluded a data science program as like in January. So that program started and has already ended. Now, if anyone is interested in doing a data science program, you would have to go through the research fellowship process, which means that if you're part of the research fellowship program, we are going to be doing the same kind of training we did as part of the data science program. And you will be going through those similar resources on machine learning and data science, and you can take that program again, because we do not have any upcoming data science program which is dedicated to it like we have for metagenomics or transcriptomics. I hope that clears everybody's doubts. And regarding all of the details, so once you fill up the form, we will be sending you email regarding all the details on how to join the program. And as I mentioned earlier, for each of the programs, we do offer scholarships. So the scholarships are offered to students who who present that they are able to take up the additional responsibility of attending each of the sessions, sharing with us updates of their progress. So the student has to be very dedicated. So once you submit one of these scholarship applications, what we do is we invite you and one of your reference to a one-on-one meeting where we meet with you, explain to you how the program works and what you would be required of you. So if you are able to agree, you are offered that scholarship. And this can either be a full scholarship or a partial scholarship. So this completely depends on like how you're coming and like presenting yourself. So that would be completely dependent. And then we would be sending you an email regarding this. So if you're applying for the scholarship position or you need help with the scholarship, you can again email us and we'll be sending you the details. So whenever you're emailing us, please mention the name of the programs, as we have mentioned quite a number of programs here. And we would love to help you with the program that you are seeking. So please mention the name of the program you are wanting to apply for, and we'll be helping you with all the details. It is either myself or Aditayo. The session is recorded, Irfan, so we will be sending you the video of the session. If there are any questions. Yeah, thank you, Aditayo. So I welcome you all to join either of the training programs and collaborate with each other. Yes, the fellowship program also has a fee. So if you go to this link, you will be able to see all the details of the fellowship program. I'm again sharing this in the email, or if you fill out the form, you will also receive an email which will have all the details.